Alrighty, it's time for us to buckle into the final topic in the enzyme series of lectures and one of my personal favorites, enzyme mechanism. Our goals today will be to first recognize that most enzymes don't obey Michaelis-Menten behavior. We're going to unpack that as we discuss the role of a prototypical catalytic triad observed in a class of enzymes called serine protease. And then we're going to apply concepts of enzyme kinetic data to the analysis of enzyme mechanism. So the goal today is to understand how the residues within an enzyme's active site work together to do the reaction they're designed to facilitate. And a great way to get the ball rolling here is with our molecule of the moment, para-nitrophenyl acetates. Para-nitrophenyl acetates is known as a model substrate. This is a compound that scientists have engineered that lends itself for the study of enzyme mechanism. And they're really good for understanding how hydrolases work. And remember that a hydrolase is a class of enzyme that will activate a water molecule to split apart a bond. And the bond that we split in our model substrate is going to be this ester. Para-nitrophenyl acetate is very convenient for studying reaction rates because when we cleave apart this ester, we're going to release a yellow-colored para-nitrophenolate compound, which we can detect through absorbance spectroscopy. So to a solution of an unknown esterase, you and your lab partner observe something interesting. You observe the generation of yellow-colored para-nitrophenolate in a burst pattern. You rapidly generate 1 mole of it for every mole of enzyme within an initial period, shown over there. So in the first couple of seconds of the experiment, the rate of this yellow product being formed is very fast, and then the rate slows down a bit. Your partner also discovers that during that initial burst phase over here, there is no acetate being generated. But after the burst period ends, we generate acetate at a similar rate to which the para-nitrophenolate is generated after the initial burst. So reproduced over here, we have the generic enzyme reaction equation that we used in our Michaelis-Menten derivation. E plus S reversibly forms our enzyme substrate complex, which dissociates into our enzyme plus products. This model does not apply to the situation we've observed, though. We have multiple products, and they seem to be released at different times. So my question to you is, how would you revise the typical enzyme model described below to account for these new data? And I want you to defend your reasoning. I also want to assure you that this is a highly open-ended question. There are many right answers to this. There are also many wrong answers. So it's supposed to be challenging, it's supposed to be a little subjective, and I trust that you will rise to the challenge. The bulk of this lecture will concern how an enzyme performs chemical reactions, and the exemplar that biochemists love to study is the serine protease. It is a very typical hydrolase whose function is to digest or break apart polypeptides, and it does that by hydrolyzing the amide backbone. This is actually a big challenge because proteins are both very diverse and very stable. So there are two problems all serine proteases must overcome. The first is, how do they discriminate substrates? Consider our digestive proteins like pepsin and trypsin. How do they know what is the myriad of food proteins entering our digestive system versus the good proteins, such as the proteins in the stomach or themselves? How do they only digest the food proteins? The second challenge they must overcome is how do they break one of the most stable groups in biology? Amides are stable by design, way more stable than esters and anhydrides and so on and so forth. And that's important because if our bodies are going to go through the efforts of making a protein, we would like to have them last a long time. So the way biology addresses this first conundrum, we've actually already been exposed to: spatial and temporal control, aka propeptide cleavage. We'll generate the digestive enzyme as a proenzyme, release it into the lumen of the digestive tract, then activate it. Additionally, our entire digestive tract is coated in mucus, which not only helps lubricate the passage of food down the alimentary canal, but that also forms a protective layer that prevents the proteases from acting upon our digestive lining. More importantly than mucus or propeptide cleavage, though, is the design of the protease active site itself. Remember how a few lectures back I said that the binding site and the catalytic site are technically two different subcomponents of the enzyme active sites, and this diagram should explain why. Over here, where we have S1 pocket, that's the binding site. And on the other end, this whole complicated mess in this diagram of a serine protease active site, we have a catalytic triad, which are 3 reactive residues that constitute the catalytic site. This is near the binding site, but it's not in the same exact location. They have 2 different jobs. The catalytic site does the reaction. The binding site recruits our substrates. And enzymes can be promiscuous or specific. Digestive enzymes tend to have very promiscuous binding pockets. Great examples of those are pepsin, trypsin, and chymotrypsin. Their job is to digest a myriad of incoming proteins: the gluten in your pancakes, the actin and myosin in your bacon, the casein in your yogurt, so on and so forth. How are they able to be promiscuous, recognize a variety of different peptide sequences? The answer is their binding site design is very simple. In the case of trypsin, it's basically a small, narrow divot filled with negatively charged aspartates. So the trypsin site looks something like that, full of negative charges. So any peptide that has a positive slender residue, like a lysine or an arginine, is going to insert inside that small, narrow, negatively charged binding pocket, where the catalytic site right next to it can come in with the scissors and snip it. So the recognition sequence, which is signified as the residues and a down arrow where we will cut the bond, is any K or any R, and we're going to break the amide bonds right next to it. If a protein has 10 lysines and arginines, it's going to get chopped up into 10 pieces. The same logic applies for chymotrypsin. It has a slightly different binding pocket design. It's a very large binding pocket that's filled with nonpolar residues. That means chymotrypsin is going to bind any residue that is large and hydrophobic, such as phenylalanine, tryptophan, or tyrosine. Its recognition sequence is F/W/Y, and then you cut the bond right after that. Anytime we see one of those residues, we're snipping that peptide right there. So hopefully you can appreciate why having promiscuous binding site suits the design of the digestive enzyme, whose goal is to chop anything down into small pieces. Now let's take a look at thrombin. We talked about this last lecture. It is a protease, but it's not a digestion protease. It is a blood clotting protease. We do not want this to be promiscuous. We want it to only cut a peptide segment within fibrinogen. So this is a specific enzyme. And therefore, its binding pocket is not going to be simple. It's going to be very intricate. Its binding pocket is going to only allow the sequence LVPGS. And the catalytic site is aimed right over here, between the P proline and G glycine. Not many proteins in the bloodstream happen to have this specific sequence, but the propeptide segment within prothrombin does have it. And that's how thrombin knows to cut there and only there to initiate the clotting cascade. So biology uses many tools to help train our serine proteases to digest their target substrates, but we still haven't addressed the central challenge here, which is actually doing that chemistry. I mentioned before that the amide bond is very, 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 very stable. But that is where our catalytic site design comes into play. Many proteases contain something that's called a catalytic triad. These are 3 amino acid side chains that are locked and loaded into a very special position that is primed to destroy whatever amide is fed into it by the nearby binding pocket. The 3 key players of most catalytic triads are, first and foremost, serine. The role of serine is to serve as the key nucleophile for attacking the amide bond. The next component of the catalytic triad is histidine. The job of histidine is to increase serine's nucleophilicity through a combination of effects. Aspartate, the third player in this triad, its job is to help orient histidine so that it is near serine and it's able to do its job. You can see our three players positioned to each other in the following schematic. We have our histidine serving as our activating group. Through hydrogen bonding interactions, it is going to interact with serine and help it attack the amide bonds. And this aspartate, also through intermolecular forces, is going to help histidine facilitate this nucleophilic attack. If that doesn't make sense now, that's totally okay. There's a lot of information going on in this slide. What we are going to do moving forward is walk you through the steps that the catalytic triad performs to hydrolyze the amide bonds in our substrate. We will now go through the 7 steps of the serine protease mechanism one by one together. I don't want you to panic because I will never ask you on a test to just draw me the mechanism from memory or from scratch. No. Instead, I want you to understand chemically what's going on in each of these steps. What roles are exhibited by the catalytic triad components? Where and when do we see, for example, acid-base catalysis, covalent catalysis, catalysis by approximation? In addition to that, I want you to be on the lookout for the nucleophiles. Which atoms are acting like a nucleophile? Which ones are acting like an electrophile? What are the leaving groups and why? If I gave you the entire mechanism as a figure and also supplied you a slightly different substrate structure, can you predict what kind of reactions would happen to it if you were to subject it to this mechanism? These are the kinds of questions I want to train you to tackle. So now that I have laid out the groundwork, take a deep breath, stretch out your fingers, and let's begin. In the initial state of the serine protease, it's going to first accept a substrate bearing an appropriate recognition sequence that complements the binding pocket adjacent to our catalytic triad. This over here is going to be our hydrophobic binding pocket. In this model peptide, we have a generic sequence that contains a phenylalanine. Phenylalanine is very hydrophobic, so it's going to dock itself very nicely in that yellow-tinged hydrophobic pocket, just like so. That is going to position the next amide bond over near our catalytic triad, getting it ready for attack. We have serine over here, a histidine on the other side of this gap, and an aspartate near that histidine. Let us now remind ourselves what the chemical goal of the serine protease is, and that is specifically to use serine to attack the amide bonds next to the binding site. There's one problem with this plan though. Serine on its own is very unreactive. It is a poor nucleophile and it is not an acid or a base. Its solution state pKa is around 14, which is pretty pathetic. This is why enzymes are so cool though. That serine isn't in solution, it's inside of a very restricted, perfectly tailored enzyme catalytic site. That nearby histidine is going to give our serine a power-up. Check out this hydrogen bond between that lone pair of the histidine and that hydrogen and the hydroxyl group of serine. And what helps histidine do that is this ion dipole interaction over there between aspartate and the other NH group on the other side of histidine's ring. This is a great example of catalysis by approximation. The enzyme fold holds these catalytic residues in a very specific proximity, which allows acid-base catalysis to occur between histidine and serine. I know serine is not an acid. However, its proximity to histidine reduces the solution state pKa of its hydroxyl from 14 to 7. Thanks to catalysis by approximation, histidine increases serine's acidity by a factor of 10 million. Which is amazing. That is what ultimately allows the histidine to deprotonate serine into ser-, an alkoxide, which looks like that. Alkoxides are great nucleophiles. The O- that we form by donating that proton to histidine is going to allow our nucleophile to attack a nearby electrophilic amide carbon within our peptide substrate, shown right over there. And that's not all. Take a look at how we have distorted our amide bond here. Doesn't that look a little bit uncomfortable? That's also by design. The protease binding pocket itself is very tight and kinky, which is going to bend the amide bonds in a very uncomfortable way, making it very strained and vulnerable to attack, kind of like flexing a stick in our hands. Think back to our stickase example a few lectures back. This increases the energy of that normally stable amide bond, making it even more electrophilic, even more vulnerable to that alkoxide we generated in situ. So I hope you can see the beauty of biochemistry all working together here. We have acid and base chemistry, pKa, enzyme theory, all working together to make this really difficult reaction possible. And the good news is, this is the hardest part of this serine protease mechanism. If you can get this down, it's all downhill from here, I promise you. So that previous step we just showed you was all prep work. Now in the second step, we're going to be conjoining our substrate peptide to the enzyme's active site serine through acid-base catalysis and covalent catalysis. And we can track that in the diagram like so. thanks to our aspartate stabilizing the histidine ring, it's able to get nice and close to that serine hydroxyl and deprotonate it. We're going to attack that proton and steal it. That's acid-base catalysis. And when we take that proton, we leave behind the electrons in that OH bond as a lone pair, a negative charge. And the resulting very nucleophilic alkoxide is free to attack the distorted electrophilic amide carbon of our substrates. This is essentially the beginnings of a nucleophilic substitution reaction. But remember, if we're building a bond between the serine oxygen and the substrate carbon, we're going to have to break another bond to make room for it. A carbon can only have a maximum of bond order 4 to it at any given time. So the bond that has to break to allow for this serine to come in is going to be the pi system and the oxo moiety of the amide bond. You see that over here. The electrons in the pi bond are going to hop up onto the oxygen as a lone pair negative charge. We call this the oxyanion. Oxyanion intermediate, which is basically the same thing as the tetrahedral intermediate. You can perhaps get a better look of it down over there. The oxygen, the O- that I just highlighted, is the same one up there. And this is the bond that we just formed between our substrate and the serine alkoxide. Now, nature is very smart. This oxyanion intermediate is hard to form because it's high energy. We're separating charge here. So to stabilize the oxyanion intermediate and make it easier to form, we're going to have a special pocket on the opposite side of our catalytic triad. And this is called the oxyanion hole. And they are formed from two backbone NH groups of this glycine and the attacking serine. These NH groups can think of it as forming a hydrogen bond with the transient CO- that forms. By stabilizing the covalent intermediate, we drive the mechanism forward. The serine protease's third step is an electronic rearrangement of the tetrahedral intermediate, or the oxyanion intermediate, where that negative charge that we formed here in the oxyanion hole is now going to swing back down. It's going to reform the pi bond in our oxo group, the C double bond O of our substrate amide. But when we do that, we have to now make a decision. Some other bond needs to break to accommodate it. And we have three choices. We could cut bond number 1, which is between two carbons. We could cut bond number 2, which is between our substrate and the active site serine. Or we could break bond number 3. which is between the central carbon and the N-terminus of the next amino acid over in our substrate chain. What's it going to be? Well, in bond 1, that's a carbon-carbon bond, which are very stable. If you break that, you're going to form a primary carbanion, which is very unlikely. So that's not the bond that we end up breaking. And bond number 2, that's the bond between carbon and oxygen. And if we break that, we're going to reform our serine alkoxide. And you know what? That's not impossible. An alkoxide can be a leaving group. It's frankly a very poor one, but guess what? If we do that, we just essentially go back a step, and we still have a deprotonated alkoxide that's just going to attack again and reform our tetrahedral intermediate. So if it does break, well, that's not going to be a permanent end result. If we break the bond between the carbon and nitrogen, well, that is going to have considerable entropic consequences. If you break that purple bond, bond number 3, that is going to liberate this entire N-terminal fragment. It's the C-terminal fragment that's held in our binding pocket over here. So chopping that part off is going to cause it to very easily diffuse away. And remember that if you go from 1 molecule to 2, that is going to be very entropically favorable because it's disordered. That's not the only reason why bond 3 is the one that we get rid of. We also have an instance of acid-base catalysis happening simultaneously. As bond number 3 breaks, it is going to deprotonate the histidine, like so. The histidine not only is hydrogen bonding with that nitrogen, kind of holding it in place, but it's serving as this vacuum of sorts. It's going to help pull apart bond number 3. And as it does that, it's going to end up deprotonating that very same histidine. That proton is going to then be ripped off and conjoined with the newly formed N-terminus of the N-terminal fragment. Think about the consequence of this instance of acid-base catalysis. The proton over here that we're now passing on to the N-terminal fragment, that used to belong to serine. We're donating the His-H proton that began on serine in the very first step. And this is important because it makes the N-terminal fragment a good leaving group. That is why of these 3 bonds, breaking that third bond is the only irreversible choice. Remember our molecule of the moment, para-nitrophenolate? This is the part that gives off the yellow compound. But we still have this part, this C-terminal fragment, stuck onto the enzyme. The rest of the steps are going to be dedicated to getting rid of this now. The fourth step of the serine protease mechanism is very similar to the first. We have accomplished our goal of breaking the amide bond in our substrate, but the C-terminal fragment is still glued on to our active sites. And we are now going to get rid of it with a second nucleophilic attack. We have one problem though. Serine is our enzyme's catalytic nucleophile. It is currently preoccupied and tied up as an ester with our C-terminal fragment. We're not out of luck yet though. Since the N-terminal fragment has diffused away from this cavern, we are now able to fill it up with water, and an incoming water molecule can serve as this second nucleophile with the help of the rest of our catalytic triad. Histidine is still held into place with the help of the off-screen aspartate up around here. And that's going to ensure that this basic nitrogen, the one with the lone pair, the one that just got deprotonated, is going to be facing this water-filled pocket. And it's going to be ready to do another round of acid-base catalysis with one of these incoming water molecules. And look at that. Water has a lot of similarities with the side chain of serine. There's an OH group that's normally not a terrific acid. However, thanks to catalysis by approximation, the basic part of histidine is going to be held close to the water pocket, and that's going to facilitate its acidification. Which is to say, we're going to then facilitate acid-base catalysis. That is going to convert water, which is a terrible nucleophile, into hydroxide, which is a good nucleophile. And now that we have a good nucleophile, this can attack the electrophilic ester carbon of our covalent intermediate. The steps you'll see moving forward are going to now look a little bit familiar. Histidine's activation of water is going to induce the formation of another tetrahedral intermediate. We're adding a bond to this ester carbon. That means we have to break a bond. The easiest bonds that we can break is the pi bond of the C double bond O. When you do that, we're going to form a transient negative charge on that oxygen. This is going to be our second oxyanion intermediate. As the oxyanion gets stabilized by our very special oxyanion hole, our histidine, his-H, that we formed in the last step by activating water, is going to be hydrogen bonding with the oxygen that belongs to serine. You might be able to predict what's going to happen in the next step. What's going to happen to the oxyanion that we just formed? These last two steps, in my opinion, are the easiest. The oxyanion that we generated in the previous step is going to rearrange and collapse. Its electrons are going to fall back down and reform the pi bonds within the oxo group, just like so. But when we do that, we're going to have to break one of these three other bonds to accommodate it. And again, we have three choices. We could pick bond number 1, which is between two carbons. We could pick bond number 2, which is between the carbon and the OH, which we just installed via water. Or we could pick bond number 3, which is the carbon-oxygen bond between the substrate and the serine. So I think we can rule out that bond, the carbon-carbon bond, because we know that carbon is a pretty horrible leaving group. If we broke the second bond, the green bond, between the carbon and the OH, well, I mean, we could, I guess. It would form a hydroxide, but the hydroxide could just attack again, and then we would go back a step and then forward a step. That's not an irreversible option. The irreversible option is breaking that purple bond, bond number 3. And there's a couple of reasons why that's the way we go. This hydrogen bond is going to destabilize the purple bond, which is good. Breaking that bond, however, will also coincide with a final instance of acid-base catalysis. We are going to protonate the serine oxygen as it leaves with the help of histidine and aspartate working together. And that is going to make it a much better leaving group. In the case of the OH, the leaving group there is poor. However, a neutral alcohol is a good leaving group because it's not a strong base, it's not a strong nucleophile. And now that we are no longer covalently bound to the enzyme, the C-terminal fragment, the last half of our product, is free to diffuse away from our enzyme. It is going to leave that pocket, and in doing so, we will have regenerated our enzyme back to the original state, where we have our aspartate associated with our histidine, with a protonated serine just on the other side, and an empty hydrophobic pocket. Every enzyme mechanism must come full circle. We have to end where we began, leaving the enzyme in the state we initially found it so it can be ready to react with a new substrate again and again and again. Pat yourselves on the back, you just survived the serine protease mechanism. It's pretty cool, right? Some students might think that we only teach you the serine protease mechanism to torture you, and that's only partially true. In reality, there are medical applications of this knowledge, such as the discovery of HIV treatments. I'm going to give you a little crash course on virology. When a virus inserts its genome into your cell and it forces a cell to transcribe and translate that into a viral protein, what it actually does in many cases is generate something that's called the polyprotein, or the group-specific antigen, which is called a GAG for short. And how this GAG polyprotein works is there's usually a protease domain at the end, all folded up. And there are cleavage sites, recognition sites that the protease will attack that I've marked here with these little notches. And I'm just making this up, but perhaps this segment is reverse transcriptase once it's cleaved off and able to fold. Maybe this is a capsid protein 1, capsid protein 2, capsid protein 3. And once the protease cuts up all these areas, the proteins can fold and assemble into a virion. This GAG protein is harmless because it's not folded yet. The protease domain at the end needs to attack and chop up those sites first. So the HIV protease domain is an important drug target for managing that illness. Scientists studied the mechanism of HIV protease to come up with this drug, nelfanavir, also known as Viricept. It has a KD of 2 nanomolar, which is really impressive. It's considered practically to be an irreversible inhibition of the enzyme. And this is a picture of the drug right over here. Scientists know that the recognition sequence for HIV protease is phenylalanine proline. So each of these notches that I drew up here is a phenylalanine proline pair, and we're going to cut the bond between them. So in the molecular structure of Viricept, we included a phenylalanine-like portion shown over here in blue. And over here in green, there's going to be a proline-like portion. So Viricept is going to fit very comfortably inside the binding pocket of HIV protease. And that's not all. The most special part, in my opinion, of this molecule is right over here. What I call the uncleavable bait portion of the drug. HIV protease is going to attempt to have a nucleophile come in and attack the portion I highlighted in yellow, but there's a little bit of a problem. The yellow portion has no electrophile present. It's a secondary alcohol. And even if it did have an electrophile, there's no leaving group. Both of these bonds are carbon-carbon, and we don't like to break carbon-carbon bonds. So Virusept is going to insert itself into the active site of the protease. It's never going to leave because it's a better fit than the own recognition sequences present in this group-specific antigen, and it can't be broken. So if you ever saw the movie Hercules, a Disney 90s classic, it's kind of like when Hercules became immortal and his thread of life got turned into that impervious gold filament, and the scissors would just break on it when the fates try to cut it. You know, the string won't cut. It's just like that. None of that would have been possible without thoroughly understanding the mechanism of HIV protease. To wrap up, we've learned today that enzymes are complicated beasts. For example, they can utilize many substrates or sometimes generate multiple products instead of just one. A great example of that being the serine protease that we covered today. Additionally, many enzymes can feature one or more reaction intermediates. Examples of that include the oxyanion intermediate or the covalently attached acyl intermediates, both of which we observed again in the serine protease mechanism. Additionally, many enzymes have allosteric sites where effectors can come in and activate or inhibit the enzyme from functioning. And either through allostery or covalent modification, enzymes can also experience conformational change, which in turn modulates their activity. These 1, 2, 3, 4 stipulations are not accounted for in the Michaelis-Menten model. And therefore, most, if not all, enzymes deviate from Michaelis-Menten kinetics. Remember that the Michaelis-Menten equation is built off of several assumptions, such as a single enzyme unit binds a single substrate to form ES, which breaks apart into E plus P. The moment you have more than one kind of substrate, or you generate more than one product, you break the Michaelis model, basically. The moment your curve shifts from a hyperbola and becomes a sigmoid, that's no longer described by the Michaelis-Menten equation. There's a common saying in science: not all models are correct, but some are useful. And the Michaelis model is certainly very, very useful. And that's why it's so beautiful. Because even though I can't think of a single enzyme that perfectly fits the Michaelis-Menten model, the application is still crucial in our understanding of enzymes, mechanism, and medicine. So it is highly applicable despite its simplicity. I hope you've enjoyed our discussion on enzyme kinetics as much as I have, and I'm looking forward to applying our knowledge of this to our in-class exercises. I'll see you soon.